good morning or good afternoon, or even good evening, depending on where you are. Welcome to the Rousselot Lunch Symposium. My name is Jeff Daelman, and I'm serving as the chair of this symposium. I hope you understand it's not feasible to ship you all a lunch given the circumstances. So if you see us around the next conference, just remind us we owe you one. So what we have in store for you today is actually pretty straightforward. I'll give you a short introduction about who we are as a company. Then Jeroen Leijten will talk about the biomedical applications for gelatins. And then I will come back on that and give you an insight in how we as a company approach the clinical translation for gelatin-based or gelatin-containing therapies. And then, of course, we have time for some questions, which, by the way, you'll see in the Zoom, there's a Q&A. You can put all the questions there, and then we'll come back to them at the end of the session. So first, a word on Rousselot. We're not a young company. We're 130 years this year, and we've been making gelatin and collagen-based products for all that time. And we're not as known yet in the biomedical segment, but there's a very good chance you have all at some point at least eaten or consumed our products as ingredients in hard capsules, soft capsules, gummies, marshmallows, other products. There's a fairly good chance there. We're part of a bigger US-based group called Darling Ingredients, which is based in Irving, Texas, and they do basically anything from pharmaceuticals, where we are in, but all the way to fuel and pet food and fertilizers. Now, we're talking quite a lot about gelatin and collagen, and I realize that most of you probably know the difference. Just as a refresher, collagen is what we have in our body. It's the piece that makes up half of our skin, a part of our bones, our joints, our tendons. We are all, on average, roughly calculated about 5.2 kilograms of the stuff. What Rousselot does is we take that collagen raw material, we hydrolyze it, partially or fully, and you end up with a gelling or non-gelling gelatin. And this is, I think, the topic of today is the function of gelatin in the biomedical. Now, besides biomedical, Rousselot historically has been actually more active in different areas. Our oldest segment, let's say, is the functional ingredients, which I mentioned earlier, is the one we typically associate gelatin with. It's food, pharmaceutical dosage forms, these kind of things, which is still the bulk of our business. Secondly, we also have our health and nutrition, and this is where we supply hydrolyzed collagen or collagen peptides for functional foods. There's a clinically proven benefit of the oral consumption of collagen peptides for skin beauty, for joint health, for bone health. That's also where we're doing much more of our business now. And then last, but I think today, definitely not least, is the biomedical segment, where we are focusing on today. And that's, I would say, that the newest kid on the block. It's where we use our old knowledge and our expertise in making gelatin and opening new routes to the market. Now, if you talk about the biomedical applications for gelatin, it's quite a wide span. It goes from cell culturing and transport as simple as a plate coating. Maybe then the next more complex is a wound dressing, where it's still, let's say, something as simple as a gelatin film. You can go down to the drug delivery carriers, because after all, gelatin is collagen based, so it degrades in the body. You can control the degradation, so you can perfectly use it to deliver APIs to certain locations in the body. And because you can control the rate of degradation, you can also control the release rate. We've been seeing it used now in post-operative implants, in microneedles, all these kinds of things. We're seeing quite some things moving. But I think where we see the most, I would say, almost sci-fi or science fiction developments is in the specialty engineered tissues. And I think mostly the 3D bioprinting, where you're trying to recreate a human tissue for drug screening or for modeling reasons whatsoever. Also there we see the high uptake of gelatin, but specifically of the modified gelatins, for example, the Gelma, to name just one of those. And that was Rousselot in a really very short nutshell. Our speaker today is Jeroen Leijten, who will talk more on the biomedical applications for gelatins. Jeroen is currently an associate professor in the developmental bioengineering department at the Technical Medical Center of the University of Twente. He has a master's degree in biomedical sciences from Leiden University and a PhD in tissue engineering from the University of Twente. In 2013, he joined Prometheus, which is the division of skeletal tissue engineering of the KU Leuven. And there he worked on the micro-technological platforms for stem cell-based microorganoids and also on smart micromaterials. Then in 2014, he became a senior postdoctoral researcher at the Kadem Hosseini lab at Harvard MIT, where he worked on microfluidics, on instructive materials, and organ-on-chip. And then lastly, in 2016, he accepted a tenure-track position at University of Twente. And his lab focuses on developing novel microtechnologies with a particular interest in microfluidics, micromaterials, and even on-demand tunable biomaterials. And he uses all these combined to create three-dimensional spatiotemporal microenvironments in which he can control the behavior of stem cells. And Jeroen will present some of the novel areas of applications for gelatins and highlight the potential effects of biomaterial consistency and purity in relation to these studies. Jeroen, I think you have the floor.
So just give me 1 second to share my screen. Indeed, so we're talking today about the biomedical applications for gelatins, and in particular the newest developments that we see. Now, indeed, as Jeff already mentioned, gelatins are not only a very old product, it's a very well characterized product. It's also used in our field, biomaterials and tissue engineering, especially because natural origin, but also based on its thermal reversible properties. But most groups who use gelatin at this moment, based on literature surveys, they use modified versions of gelatin. For example, what happens if you put a methacrylic anhydride on top of a gelatin, you'll get a gelatin. And as it used to be called gelatin methacrylate, in 2017, I think, we published a study in which we actually looked at what the actual structure of that biomaterial was. And we figured out that there's actually two modifications on the backbone. So hence we had to change the name from methacrylate to methacryloyal. So if you ever wonder what the difference is one is chemically correct name and the other one is a outdated name. Now, the interesting from the gelatin methacryloyal, which I'll now call Gelma, is that it has all the benefits from gelatin, but it's also photo-cross-linkable, and it also allows for tunable cross-linking, meaning that it's not a binary switch, it's solid or it's liquid, but you can also control, for example, the amount of stiffness. As Jeff already introduced, there's a whole long list of already pretty well explored material applications. Think about general hydrogels for encapsulation or release of drugs or stimulating factors, bioprinting, microgels, microneedles, patterning, injectable materials, etc., etc. For today, we're going to look at a few of the newer areas of application, and specifically, how can we use gelatins to improve the incorporation of nanomaterials in hydrogels, which is something that is started to boom in the field, and we expect to grow exponentially over the next few years. Also, we are looking at the regenerative properties of materials such as gelatins and gelmas by specifically tuning the immune host response towards regeneration. And also, I would like to talk a little bit about the novel modifications that are available for gelatins and, for example, modified versions of Gelma, for example, to give bioadhesive or hemostasis or energy storage, batteries, in fact. Throughout that presentation, I'll also take you along some of the things that we haven't published. So what is actually bugging and challenging us in the lab. So when you work with gelatin or gelatin modification products, such as Gelma, what is actually important and what should you consider when purchasing Gelma or gelatin? I'll do that on the example that one of our projects is working towards, and that is to create electrically conductive Geoma hydrogels. Now, there's different ways you can make hydrogels electrically conductive. By nature, hydrogels are pretty good insulators in the sense that if you would like to stimulate cells, they are not quite suited for those tasks. So you would need to modify it. One of the ways you can modify such a hydrogel is the incorporation of nanoparticles. You can think about carbon nanotubes, you can think about other materials, and today, specifically, we're going to focus on gold nanoparticles. So basically what you would do then is you would take a gelatin, you would mix in your nanoparticles, and then we would use the advantage of the gelatins in order to do a 3D bioprinting, and the goal is to do that in situ or topically, so we can regenerate patches of the heart instantaneously and during surgery. Now, the big challenge for those incorporations is that putting these nanoparticles in is really simple. It's as simple as mixing it. But if you wait a few seconds, then you will see that they also start to aggregate and basically form big clumps. At the moment they form clumps, the distance between those particles, if they're outside of the aggregate, becomes extremely large, which means you still end up with a very poorly conductive hydrogel or gelatin. Now, once you are able to solve that challenge, and that's something we've been working on, and the answer is on the next slide, and the answer is actually gelatin, then you can actually use this for a very wide range of applications. So for example, you can engineer new muscles, skeletal muscles, but also, as our goal is, heart muscles. You can also start building neural integrations, not just for getting neurons to grow in to, for example, actuate your tissues, for example, for muscle and heart, and specifically skeletal muscle, that is essential, but also to, for example, make connections with soft robotics and bioelectronics. If we actually would like to integrate that and move that field towards something like cybernetics, we really need to have such a modified hydrogel. Now, as I said, normally if you take a gold nanoparticle and you bring it into an aqueous solution, you'll get very nicely the interaction between these particles and they start to cluster. Now, gelatin, in this case, very short chains, low molecular weight Gelma, has an interesting buildup. It's a complex protein with many different amino acids arranged in a very peculiar way that it's positively and negatively charged. Very often, this is seen as a negative, for example, that you will get biofouling, which needs to be prevented. But we actually use this biofouling as a strategy to prevent the flocculation and the aggregation of these nanoparticles. So in this case, we activated the surface of the golds to enhance or further enhance the biofouling, and then we added these very short chains on top of Gelma, on top of these nanoparticles. Now that does two things effectively. First, that means that these particles are no longer effectively capable of aggregating. Secondly, now on top of that surface, we have a cross-linkable moiety, which means we can now physically entangle these gold nanoparticles in the hydrogel via covalent cross-linking. Now, we generated using TEM data that indeed we can first activate the surface and then show that we modify the surface based on contrast measurements. And then more importantly, we can also show that there's a difference in the potential, in this case, zeta potential. If we then mix it into the polymer solution, then indeed you would see that these particles really stay single particles. They stay separately, and that's pretty unique. If you would not do this, you would get aggregation, and basically all those four would be in one region. Now, by just simply adding more or less of these gold nanoparticles, we can then control the impedance or the electrical conductivity, if you will, of these hydrogels. So in that way, you can get a very easy electrically conductive hydrogel. Moreover, we show that that had very little impact on the mechanical properties of the gel itself. We then use it for an advanced 3D printing technique. So we combined a gelatin with an alginate to make a complex printable bioink. We used, in this case, the alginate for immediate cross-linking. So we could deposit it using an extrusion technique with immediate cross-linking using the divalent ions of calcium. And then afterwards, we did a post-crosslink to crosslink our Gelma. We then leached out the calcium and therefore also the alginate, so we could get a very nice porous gelatin structure. The reason why this is red is not because we colored it, but that's actually the color of the gold when you mix it into a hydrogel. We then showed that this is very nicely printable, so it doesn't affect the print quality, but we can also show that it doesn't affect the viability. And most importantly, we showed that if you use cardiomyocytes, cells of the heart, that if you use a relative pristine Gelma, you do have very nice behavior of your cells within. You get the elongation, you get the stretching, you get your cell-cell communication. But once we start to combine it with the cold nanoparticles in there, you see that they also get these kind of cellular organizations, for example, these sarcomeric alpha-actinin organizations, which are very important for also the contraction and the beating of the engineered heart tissue. So not only do they get better cell-cell contact, but also, for example, beating frequency and even more, the beating strength goes up. So this is really an effective way of creating nanoparticles inside of a hydrogel network. Now, as I already mentioned, that's pretty important because normally you get this aggregation, which gelatin coating can overcome. But also if you use, if you would be able to get it in, for example, very fast cross-linking after, for example, shearing it, then you would still very fast lose your nanoparticles because they're small and they would simply diffuse out of your network. If you actually use this trick with Gelma with low molecular weight, it's physically cross-linked in, which means this is stable over the duration of the print, of the biomaterial. So we wanted to move this forward, and we wanted to move this towards animal models. Specifically, we wanted to know not if these cells then actually performed in terms of cardiomyogenesis and survivability, but we also wanted to know how the host would start responding to our gelatins and our modified gelatins. So for this, we did quite some research, but what I would like to talk about today is the macrophase polarization. And specifically, we are going to simplify this towards M1 and M2. For those interested, yes, it's much more complicated in the terms that there's, for example, already 3 M2 phenotypes, and also these are plastic, which means they can switch through it. If you'd like more information about that, please do contact me. But for now, for the duration of this talk, we'll simply we'll talk about M1, which is pro-inflammatory, and M2, which is anti-inflammatory and pro-regeneration. So we used Gelma, as that was the material that we wanted to move forward towards clinical application. And we used another acrylate modified material, which is, at least back then, very popular and very commonly used. So it's a polyethylene glycol with a diacrylate modification. Now, the first thing that we did when we started to create these constructs, which are basically these gels in which we in 3D encapsulated monocytes. We did this with human monocytes as well as with cell lines, and gave the same results. But what those results are is if you put these monocytes inside of a synthetic material such as PEGDA, you will get a very strong M1 phenotype. If you would add IL-4, which is a very potent inducer of the M2 phenotype, there's hardly any benefit. So, which means that once you even give strong cytokine stimulation to become M2, in such a synthetic material, it simply does not respond. In contrast, if we would look at a Gelma, then we would see very little stimulation. It's very little activation of M1 markers. It is there, but not so much. But once we add the IL-4, you can see that these cells are really responsive to this. So at the moment we add it into a Gelma, you see that these cells respond by becoming basically negative for M1 markers. Similarly, PEGDA had close to 0 M2 markers being present. In this case, we report on CD206, but this is also true for other markers at the immunofluorescence level. But also then if we look at the stimulation for M2 markers, in gelatin, your cells are actually sensitive. So in other words, we had some evidence that in PEGDA, you would drive your pro-inflammatory macrophage polarization. Well, in Gelma, you get pro-regenerative polarization, which was for us very reassuring. Now, if we then look at what is not expressed by the cell, but secreted by the cell, so we took off the supernatant of these cells, and we did these kind of dot plots where we see all these cytokines, which we can then quantify. The interesting thing here, which I can leave you with, is that if you then compare PEGDA with, for example, Gelma, then what you will get is if you add IL-4 to stimulate your M2 pro-regenerative phenotype, you see that you have both M1 and M2 markers. Now, that would suggest that you still get some M1 differentiation. However, we also realize that in vivo, it's much more complex. It's always a mixture of pro and anti-inflammatory cytokines. So we then added in LPS, lipopolysaccharide, a very common and very potent used molecule to induce inflammation and also drive M1 polarization of macrophages. And then something very interesting happens, and that was not reported before in literature, and that is that you get a quick, very black and white picture of how different materials work. So if you give a simple stimulation, you get a bit of a gray, murky area, M1 and M2 is upregulated. But if you then look at a more biophysically relevant scenario where you have pro and anti M2 activation, you'll see that you have very potent M2 activation and very little M1 activation. While, for example, for PEGDA, now they're activated to become M1. Well, here in just the IL-4 stimulated, they're not. And you still hardly have any activation of your M2. So M2 macrophage polarization is very dominant in Gelma hydrogels, which makes sense as they are natural materials and they're used quite commonly for a variety of applications. We still wanted to know why this was, and we figured out using a lot of blocking techniques and blocking inhibitors and knockdown assays that it came down to integrin presence. So can we stimulate the right type of integrins? Specifically, if we would block specific integrins, then for Gelma, which is stimulated to become M2, then if you look at the M1 marker, it's hardly there. If we add an inhibitor for the integrins, you see that it fully becomes M1 and it becomes as strong as your, for example, PEGDA. Similarly, for your M2 markers, under natural conditions, you would get a very nice M2 phenotype. And once you block that integrin binding, then you would go fully back to a negative phenotype. So now this is a very strong M1 phenotype. In essence, if you want to stimulate your cell, you would need to have an integrin interaction. In this case, Gelma achieves that by having a correct density, because the concentration is also important, of RGDs being present within your biomaterial. If you do not have this, you end up with a pro-inflammatory and pro-apoptotic phenotype, an M1. Well, if you use Gelma, it naturally has the right concentration of these molecules if you add IL-4 there as well to get a M2 phenotype, which means it's a very nice material for pro-regeneration. Now, this is something that is unpublished. For follow-up research as well as answering reviewer questions, they asked us to do a couple of other experiments. Now, at that moment, our batch that we were using for the entire experiment was finished. And the first follow-up study that we did is we did not got M2, we got actually an M1 macrophage polarization, which surprised us very strongly because over a 2-year course of time, we never saw that. Now, the only thing that we changed was the batch. So we then actually took a new batch of, we bought a whole different set of batches of gelatin, modified it, and then tested it. And in total, we tested 7 batches. 4 of those batches gave us M2 polarization, and 3 of those batches gave very clear M1 polarization, which means there was batch-to-batch variation, how this would stimulate the outcome of your immune response. We then did a whole lot of different experiments, and basically what we found is depending on the supplier, as well as for some suppliers, sometimes you get it, sometimes you don't, and some suppliers never have it, you will have very potent LPS contamination. Now, if you have a lot of LPS inside of your system, you drive M1, which for me the take-home message was, and then we start switching suppliers, is we really need high-quality gelatin, because otherwise we get reproducibility problems. So although everything in that first paper was very accurate and completely correct, we know that if you would try to reproduce this with contaminated versions of gelatin, you would get a very negative response in terms of immune cell polarization. So that's one. It's regenerative, and we can use it if it's a clean material. We also then looked at how to apply that material, which we now have in a clean version, towards the heart, because we had an electroconductive, clean material at that point. But we still needed to apply it to the heart. And the heart, you cannot simply place it on, it needs to be bioadhesive. So therefore, we started to explore also additional modifications of gelatin. In this case, we used a thiamine that I spoke earlier about in the Jean Rey Award. And also, I was happy to see that Ricardo Levato, also this morning, was talking about these type of modifications. And then basically you can modify your gelatin to have a thiamine unit. This is very simple chemistry. It's type of NHC EDAC reactions. So pretty straightforward. And basically what you then get is a gelatin backbone with these kind of monophenolic compounds, these thiamines on top of the backbone. In the presence of a catalyst and an enzyme, you can now create hydrogels. The interesting thing there is that if you apply it to a tissue surface, for example, a heart or a cartilage piece, then you will have natural matrix sticking out of the surface. It's pretty rough, these type of tissues. And these kind of tissues have matrix, and the matrix, for example, fibrin and collagen, will present also monophenolic compounds that are analogs of thiamine. They're tyrosines, which means that if you now apply the GLTA on top of your tissue, you get a very nice strong bond. As Ricardo already reported as well, you can increase the bonding strength of the material with over 15-fold, and we're able to do it by 50-fold. Now, not only can you then bind it to the tissue itself, but you can also start then binding it to cells, we realized, by binding these thiamines to tyrosines, which we then demonstrated with mass spec analysis that indeed happens. So if you look here at this little peak, you see that if you mix thiamines and tyrosines together and then react them, you get indeed coupling at the molecular level. You can use this to coat Gelma on top of the surface of a cell to, for example, create exocytoskeletons. Or, for example, like we said, you can then also start binding in your cells into biomaterials. This is really at the cell surface, as we demonstrated using confocal microscopy. And also, if we analyze it, you can see it's outside of the cytoskeleton, the stained actin. You can see that the thiamines are really outside. So that means that it's inside of or on top of the lipid membrane. Indeed, if we would then put cells inside of a hydrogel, in this case a single cell microgel, you can see that the cell membrane is really glued to your material. Now, the interesting thing there is that we show that this, again, operates via the integrin, so you can very nicely stimulate the immune system with that as well. We also, in other work, showed that you can indeed use this to get an M2 response, and if you implant it in mice, you get a reduction of fibrous capsule formation. And then basically we wanted to know how does that work, for which we took the cells, labeled them with a thiamine, and then did a pull down assay followed by a mass spec. We overlapped it with the meta-adhesome that was published previously by others in literature. And then we could indeed see that these thiamines bind to fibronectin and collagen, as I said on the previous slide. And specifically, it operates over this axis, meaning it operates over the integrin couples of alpha-5 beta-1 and alpha-V beta-3. Indeed, this axis of integrins is known to stimulate and control to a certain extent the immune responses and the macrophage polarization. Also interestingly, if you then make materials of different stiffness, you can start using this thiamine tyrosine, so the cell material coupling, you can start using this to also start steering cell fate, for example, steer it into fat production, adipogenesis, or bone production, osteogenesis, and you can do that in a very controlled manner. Now, that effectively means that if you start working with new modifications of gelatin, for example, GLTA or thiamines conjugated to gelatin, you actually offer model and novel modes of mechanotransduction. So if you have your RGD, that is one mode via specific integrins, and your thiamines are stimulating it via a different section. And indeed, we're now starting to find that you can steer cell fates in completely unique ways that were impossible to do before, or at least we were incapable of doing so before. So that means we were looking at how can we modify these materials. The GLTA was very satisfactory, but we also wanted to have a system that would both do adhesion, like the thiamine, and electrical conductivity. So for that, we started to look into a completely new modification of gelatin. So we started with a Gelma, and then we incorporated a bioionic liquid. and in this case, a choline acrylate. The advantage of that is that it will have the same type of, or it would also offer bioadhesion as we just discussed, but also choline acrylates are a bioionic liquid, which means it's also electrically conductive, which means for the hard application, that would be amazing for applying it and then having functional performance. So we first wanted to make sure that this new modified gelatin would actually work as we would say. So we took this biogel, as we call it, bioionic gelatin. We placed it between two pieces of tissue, cross-linked it with visible light, and then basically start doing lab shear test, tensile testing, and burst shear testing, using putting the hydrogel between a plate and an orifice, and then putting pressure on it from below, and then seeing when it would actually fracture. Now, as you can appreciate from here, already gelatin has pretty interesting properties, but if you would include a bioionic liquid, you can very nicely control the shear strength of the material, the adhesive strength, as well as the burst pressure. So that means that we have a new type of bioadhesive that we can use. More importantly, if we would apply it to such a tissue like the heart, you are always working in an environment that is probably covered in blood. So having an open wound, we were also looking at whether we could make gelatin into a bioadhesive hemostatic hydrogel, meaning that if we would apply it to the tissue that we would be interested in, would it then also help with the blood clotting? And indeed, although gelatin by itself has really nice properties to prevent blood clotting, or at least, let me say this more accurately, it has zero effect or very little effect on blood clotting. Once we start to include this bioionic liquid, we can very nicely tune the speed of blood clot formation, the formation of the blood clots, and including the network formation and the cellular infiltration within these networks. And this can be done in a near linear fashion over the concentration of the bioionic liquids. Indeed, if you then actually take hearts, in this case it's a pig heart, and you do a traumatic injury, in this case you really make a hole inside of the heart, for example an impalement, then we could very nicely coat this and close this in a functional manner. You can have severe lacerations where we show that you can close those up. And for example, one of the worst and difficult assays that you can do is a liver puncture. Once you puncture the liver, it will start to bleed uncontrollably. And what we show is that this material can even close up those wounds and prevent massive blood loss. So in this graph, we basically show that we can patch up hearts and lungs and tails and livers, but also doing so in a life-saving manner by reducing the blood loss of the patient. Now, that comes to one of the challenges, and that is, this is an amazing material, but then if we would move this forward, like the Daelman modified materials, we would like to clinical and preclinical testing, but then we need specific high-end formulations. So for that, we would need partners in order to help us, because in every grant that I've written, I say we can do that, but in reality, it's our private partners and collaborators who are going to actually do that for us. As a final application, also using these bioionic liquids, I already said that we can very easily now conduct electricity through this bioadhesive material. So we now are also using this to create implantable batteries based on gelatin, which means it's a green energy or green chemistry approach for which we can actually make biodegradable batteries for implantation purposes. And then you can think about biosensors, diagnostics, biorobotics. All these kind of elements would require a electrically conductive material as well as an energy source. So we again made our Gelma. We modified it using the bioionic liquid, meaning bioadhesive and regenerative. And then we basically made an interdigitated material based out of also an electrically conductive material, but with oppositely charged. So in this case, it's a leponite with a graphene hydrogel. And then we use bioprinting to create these interdigitating micro supercapacitors. Now, these have about the same energy density as you would have in a traditional battery, but they also have the charge rate and the relaxation rate of supercapacitors, which is very nice. So we just showed that indeed these gelatin-based batteries are highly functional, And then we can print this on top of the skin. And this is really stuck because it's an adhesive. We also did that we can implant it under the skin or in other locations of your body. And then basically we connected it in a very crude way towards a offside. Here you see not connected. And here we connect it to the on-skin printed gelatin battery. And then you can see the light switches on. So in this case, what is implanted beneath the skin is actually powering a battery outside of your body. Obviously, our end goal is to do this inside of the body, but for visual purposes, we showed it this way. Again, when we were starting to print here, we had batch-to-batch variability. In this case, the molecular weight and the viscosity were very important assets that were not always stable depending on where we bought our materials. And normally it just means your print is a little bit thicker or a little bit thinner. But if you would create these very nice interdigitating structures where you need to control the distance between these elements, you can imagine that then viscosity really becomes an important element. Also, if you control rasters, for example, to control fluid flow through these 3D printed structures, having a batch-to-batch consistency has in the past been a problem for us. So we always publish everything using a single batch of material. In short, I would like to leave you with these messages. First of all, gelatins are really an ideal material to incorporate nanomaterials inside of your hydrogel. They can not only make sure that they stay separate, but they also allow for the retention via the physical cross-linking. The gelatins can steer your immune system in implantation studies or in clinical translation towards a regeneration, or at least for the macrophages as we've tested to date. And I've told you about new modifications of gelatin, and specifically I told you about bioadhesiveness on both tissue and cell level. I talked about making a gelatin-based material that is in hemostasis, or at least it has hemostasis properties, and even that we can endow it with energy storage capacity. The major considerations, as for the unpublished lessons learned that we have incorporated in my lab, is you really need to choose your gelatin really carefully for 3 specific reasons. The first is the purity. If you have LPS contamination, which in our experience, now we are testing for it, is actually pretty commonplace for different vendors, you really need to be sure that you have a vendor that does not have this. The reproducibility is very important. You have batch-to-batch variation, which actually requires us, for if we do not have this, to do a lot of experimental re-optimization. So this leads to time loss. And also the production quality. If you actually want to go towards a clinical translation, which many of us do, you really have to think about starting with a good high-end grade of material. Because as I just said, if you do not use such a high-grade material, your material might actually behave differently, which means that at the moment you enter preclinical testing, and to go towards translation, you have to do a lot of extra work or work that you already have done. So with that, I would like to thank my lab members as well as the funders of my work, as well as Jeff and Rousselot for inviting me to talk about gelatins, which is, in my opinion, highly interesting. And with that, I just want to say, if you're interested in bioprinting and you are a postdoc or shortly becoming a postdoc, do feel free to reach out. With that, I would like to give the word back to Jeff.
All right. Thank you, Jeroen. It was really interesting to see all the wonderful opportunities with gelatin, all the really nice stuff you're working on. It's really amazing just the opportunities that are possible. It's mind-boggling. Which, by the way, a small reminder, there's still time for some questions at the end. So if you have questions, put them in the Q&A chat box. If we don't have time, we'll answer them anyway by email. But if you have questions, put them in there. Just for the remainder of this symposium, I'll briefly kind of respond to the items Jeroen threw up in terms of how we as Rousselot can respond to these kind of things in terms of clinical translation and consistency. So maybe first importantly, why do we as a company go into the biomedical? Well, simply because we believe we can make a difference. We want to, our darling motto is we create solutions that sustain life. We believe as Rousselot we can do the same thing in a biomedical fashion. Now, we make gelatin, so it might be a bit big of us to claim we can really make a difference, but we can, and we will by removing a hurdle or a barrier. And I'll use Gelma as an example. Gelma is not new; it's been around since 2000 actually, and it was invented at Ghent University. But since invention, there's been a lot of research and applications, drug delivery, microneedles, adhesives, cell encapsulation, bone reconstruction. The list goes on in terms of patents and publications. Yet to our knowledge, and if you know one, please let us know, but to our knowledge, none of these applications have made it into human patients. And the question is, why is that? Is there something wrong with Gelma or gelatin? But no. The reason is simple. There wasn't enough Gelma available, and there's plenty of suppliers able to sell you a few grams, maybe a few tens of grams. But if you need a few kilos every year for a medical device, that's not enough. Additionally, what was available was not the right quality. You get a lot of variability, a lot of impurities. If you want to put it in a human, it has to be clean, it has to be high purity, GMP Gelma. And that's what we're doing now. We're launching a GMP grade Gelma early next year. But there's also, and I think relevant for today, we believe we can also make a difference at an earlier stage. So the GMP Gelma that plays for the, let's say, unlocking the clinical trials, but also when it comes to R&D, to scientific research, it is, and as you already highlighted, very important to use high quality, consistent materials, because variability is important. And when I did my own PhD, and it's quite a while ago now, I did it on predictive microbiology of bacterial spores in food, so completely out of this field. I spent a few months at the end of 2011 in Nantes, in France. And you may know, you may not know that if you're working on foodstuffs and bacterial spores, variability is a nightmare. Strain to strain, batch to batch, it's all really a nightmare. And my French promoter, Jean-Marie Momret, she had a very nice saying, and it kind of stuck to me. She said, variability, she says, is the curse of the modeler. There's always variability. You can't live without it. You have to live with it. But at least you can reduce it. And if you're talking about variability, I want to work on two topics today. One is the molecular weight, let's say the physical-chemical consistency. And the other one is then the impurities, anything that's biological or chemical impurities in the product. I will start with the physical-chemical one first. And there's a lot of an important point. First, at Rousselot, we don't use bloom if we're talking about Gelma. And there are quite a few reasons why bloom is just simply the wrong characteristic for Gelma. Because, for example, the bloom of a Gelma also depends on the degree of modification. The higher you modify a Gelma, the lower the apparent bloom becomes. So it's not an independent parameter for the gel strength or of the chain length of the gelatin. And additionally, you can't really measure it well. It's too, let's say, inaccurate. So if you want to, let's say, bring this to a GMP product, you cannot really measure the Gelma, the bloom correctly. So, what to use a parameter. So we use molecular weight instead of bloom. And you'll see that representation, we always talk about molecular weight, not about bloom. And actually, there's one additional thing wrong with the way it's sold here or presented here. What they mention on these labels, that is not even the bloom of the Gelma. It is the bloom of the precursor gelatin used to make the Gelma. And I spent quite a long time with colleagues looking for a good analogy because it's so rare to do that. It's like describing the mechanical properties of glass by testing the sand it was made from, or describing the stainless steel by giving the specification of the iron ore. There is a relation between the precursor and the bloom and the Gelma, but it's not a very good one. And to give you an idea, I'll give you some data. Say you start from a 300 bloom gelatin, standard one you can buy pretty much anywhere, and then you start modifying, you add acrylates. Well, what you get in the end? It depends. It depends on the time you took for the reaction, on the reaction temperature, on the pH of the reaction. It probably even depends on the PhD student doing the reaction. And I'll prove it to you. We bought 8 commercial, let's say 300 bloom Gelmas, and we tested the molecular weight of the end product. So you see on the left, that ranges from around 130 to 160 kilodaltons. So it's anywhere from 260 to 300 bloom, let's say, in gelatin equivalents. By contrast, our own product, which I put there for comparison, is nicely grouped around the 160, 170 kilodalton mark. So probably all know how important the stiffness of a gel or a bio ink is for your cells. So if you see this kind of variability, it probably has a big impact on the output of your model system. Same thing applies if you look to the degree of modification, where we see some results. Again, the same 8 commercially available 300 bloom 80% Gelmas, where you see that the degree of methylation goes from 65 to 100%. So it is kind of a guess what you're going to get. If there's 2 things I would like you to take away from these 2 graphs, that please don't use bloom for Gelma. If you make your own, measure molecular weight after modification. And if you buy Gelma anywhere, then ask them for the molecular weight post modification. And maybe word of caution, being aware that in general, if you make it in-house, there's going to be variability. It's impossible to prevent it if you make it in-house. And that was, I think, the physicochemical aspect, but there's the same thing when it comes to endotoxins or other impurities. But I'm specifically talking about gelatin, which is a biomaterial from a natural origin. There's going to be some bacteria, there's going to be some endotoxins. And you might say, endotoxins, yes, it's regulated once I go to the clinic, that's important. But why would it matter in vitro? This is some data we've got from 5 different Gelma samples commercially available. You can see the endotoxins levels, and that's a blue bar. It goes from, let's say, slightly less than 100 all the way up to 90,000 endotoxins per gram. So our product is below 10. And now you might say, yes, is that an issue? 90,000, how much is that? Does that cause any reaction in the tissue? There was a study in '94, and they wanted to create a model system for inflammation in a joint and use the horse. And they said, okay, we'll inject LPS in the joint, and we'll see what dose causes inflammation. Well, actually, I'm going to show you 2 numbers. 1.25, which is the lowest they tested, that was enough to cause inflammation in a joint. And even if they would repeat for, I think, at least 3 days, and it would repeat it, it would stay inflamed and stay inflamed. They also tested higher doses, and actually 5 endotoxin units was enough to cause systemic inflammation in the horse. So they got fever, they got depression, they had to pull the horses from the study. So just to give you a bit of scale, 5 endotoxin units is enough to cause inflammation in a horse. So imagine what it does if you put it in a cell culture. What do we do differently then is that Rousselot, our end goal is to bring products to patients. So our entire setup is designed to have purified products that are low in endotoxins, low in impurities, which have a controlled production process, and which in the end will be available in a GMP grade. But it's also scalable. So what we supply now as a research grade, and we'll make a few, let's say 30 grams or 100 gram batches. If we make that same thing in our pilot scale in our factory, in a kilograms or 100 kilogram scale, that is the same physical product, it just has more paperwork attached, which is important because it means you can take your research and you don't have to repeat it with a new Gelma. You can just use the old research for that. And there's more. But I think Jeroen already mentioned on the in this last slide somewhere on the gelatin tyramine. And as he said, it's made using EDC and HS chemistry. And we had a lead for that saying, we need gelatin tyramine. We make about 8 grams a day in the lab, but we need a few 100 kilograms per year eventually if we go commercial. So they asked us, can you do the scale up and do a validation of a GMP quality? And we said, sure, we'll give it a try. The honest answer is no, we can't make gelatin tyramine reliably, consistently, pure. That's not possible. Just as a general comment, EDC and HS chemistry does not scale well. But we can make this. So we've launched recently a gelatin desomaterazine, so not tyramine, but a desomaterazine. It has the same functionality. If you look at the molecule, you end up grafting phenol groups to the gelatin. The only difference is that they're on different locations. They're attached to different amino acids. And this we can purify, we can control the consistency. So we have a product we can bring to GMP. So what are we offering currently? It's a range of purified gelatins, can be gelling, can be porcine, bovine, hydrolyzed, high bloom or low bloom, but also 2 types of modified gelatins. So one well-known Gelma, but also in various molecular weights and degrees of modification. And then, of course, our most recent is the gelatin Desomaterazine or phenol modified gelatin with a similar, let's say, customizable nature where you can choose the molecular weight and the degree of modification you want. And importantly, we can supply all these in the research grade, but also in the GMP grade. So there's always that route open towards clinical translation. And I think more importantly for today, we're making Gelma, we're making Gelma, but if there's another biomaterial what you're working on, and you have the aim of going towards clinical translation, and you're not sure how it will translate, and if there's any limitations, or you want to have to see if it can be purified, by all means, contact us. We're always willing to listen and to think together with you if we can assist you with any of the challenges you have. And I think that's it for me. I'll take a quick look if there's any questions in the Q&A. There's one question which asks, could you please explain the reason molecular weights difference between Gelma and gelatin? So yes, I presume when you start with a certain gelatin, why do you end up with a different molecular weight in the Gelma? It's because of hydrolysis, specifically so thermal and acidic. Gelatin is very sensitive to the combination of both. So if you have a temperature, let's say above 55, and the pH is acidic, you get a very fast hydrolysis of your gelatin chains and hence reduction in molecular weight. And then of course, that's why time, pH, and temperature all play a role in that. And that was, I think, unless there are other questions. All right. There's one more question. How is the gelatin Desomaterazine cross-linked? It's the same method of cross-linking like the gelatin tyramine. So you can use the enzymatic route using the horseradish peroxidase and H2O2, but you can also use certain cross-linkers. Not all. I think, for example, that LAP doesn't work, but there's a new ruthenium cross-linker, which I believe does work with the gel dot. But yes, and there are other ways as well described in literature. In short, anything that works for the gel tyramine also works for the gel Desomaterazine. A follow-up question on that: are there any of these cross-linkers injectable in clinic? That's a good question, and that's also a challenge with Gelma. Many of these photo cross-linkers or other cross-linkers are not available in a GMP grade. So that's something we're also looking for. If you know anybody who can provide them in a GMP grade, we're always looking for partners in that sense. But that remains a challenge. That's true.
And also a small addition: the first tyramine-based material that goes into clinic will go into clinics in 2022. So upcoming year, the first material will enter a phase 1 clinical trial. So they are coming.
There was a question: which sterilization method do we use on Gelma? Well, the Gelma we supply is not sterile as is, but it can be filter sterilized. So you can just pull it through a 0.22 filter and then you get a sterile solution. In our case, doesn't work with all Gelmas. Again, has to do with the impurities in the product. All right. And I think if there's no more questions, I will say thank you, Jeroen, for making time for us and for giving a really interesting presentation. And I would thank you all for joining us. And again, I still owe you all a lunch at a later time.